Hi, I'm Charvi Narayan. I'm a senior editor at Nature Neuroscience. We are at the 79th Cold Spring Harbor Laboratory Symposium on Quantitative Biology, and the CS topic is Cognition. And today I'm joined by Alex Pouget, who's at the University of Geneva. And Alex, you'll be talking about how the brain actually encodes information, right?
Yeah, that's right. I'll talk about some recent work that we've been doing over the last 4 or 5 years now, trying to really understand how the brain encodes information.
Okay, so this is from quite an abstract computational perspective. So are you sort of comparing the brain to how a computer works? Is that the sort of approach that you're using?
Yes, indeed. There are some interesting kind of analogies that you can draw there. I mean, so basically, any kind of computing device, the first thing you need to figure out is how does the device encode information. Mm-hmm. So if you want to understand how a computer works, you've got to understand first that it encodes information through those strings of bits, binary elements that can be 0 or 1s. Unless you understand that, there's no way you can then understand how it actually computes.
So how do you approach this problem with respect to the brain?
So with respect to the brain, of course, we have to rely extensively on recordings from our colleagues who are doing experiments in animals and presenting a variety of stimuli or asking the animal to move, and at the same time they record from the neurons, and then we try to establish what the mapping is between the stimulus and the responses that they see.
And when you do try and establish these sorts of mappings, so what do you find? What are sort of the interesting things that emerge from the sort of computational modeling that you do?
So there are quite a few principles that are emerging, but currently the main hypothesis that we're pursuing is that the code in the brain must be redundant. Uh-huh. And perhaps here I should give you a little bit of background for why we're pursuing this. So there's something very interesting about modern computers. So they encode information with binary strings. So if you have 64 bits, you can encode 2 to the 64 different numbers with absolute reliability, with no errors, which is a staggering number. I mean, it's like billions of numbers that you can encode. But for this precision with a computer, you have to pay. It comes with a cost. There are 2 different costs that you have to deal with. One is your bits have to be incredibly reliable. They're not allowed to switch. If it's a 1, it cannot switch. And the way we do that with electronics is we use electricity and we use voltage. We use about a volt to encode a bit. And as a result, you need a lot of power for your computers. And today, the supercomputers that are running in the world, the single supercomputers usually cost of the order of $2 million just in electricity to run this whole thing. So that's one, it's a power issue, right? And the second issue is reliability. If any piece of hardware breaks in those computers so that the bits can no longer be encoded, you throw away the whole thing, or at least whatever, the board that broke, that's gone. You can't, so it's extremely non-robust to damage. And the contrast with the brain is striking, because it's exactly the opposite with the brain, right? We survive, in the US, you can survive on a few $1,000s per year to just feed your brain if it's all you need. And your brain is incredibly good at handling damage. So I unfortunately lose something, I think, of the order of 30,000 neurons per day right now at this point in my life, and it's only going to get worse. And I'm told I continue to function reasonably well. It's called graceful degradations that we see in humans, that you can, by the time you're 80, you're probably at half the brain weight that you had when you were younger. And
but
yet you
even
continue
at that
to function.
stage, I assume the amount of data that the human brain is handling far outstrips any supercomputer.
Yes, and on top of that, yes, in terms of performance, we're just absolutely incredible. So we do it for very little cost, very little energy, and we're incredibly robust to damage. And so that's one of the big questions for us, is what's so special about the neural code that allows us to have these properties?
And what is so special about the neural code?
So we think the key is redundancy. That is, instead of having a bit that's infinitely reliable, what you can do is you can have multiple copies of the same bit, but they don't have to be perfectly reliable. And if you have that, then you can know the values of a bit not just by looking at one of them, but just by taking the average of the multiple copies that you have. And the fact that you have multiple copies is what we call the redundant code. This is where the redundancies come from.
But one sort of obvious question which comes from that is, wouldn't you need a large space? Then the brain is also quite compact. But if you have multiple bits of information, that does not create space constraints?
So it does. So you do end up having to have more neurons than you would if you were more efficient. But again, if you want to be robust to damage, okay, it's got to be the case that you can get the system to function reasonably well, even when you've lost half of the component in the system. And that's the way our brain works. So yes, we pay that price. We have way more neurons possibly than we could need in principle. But again, that means much less energy. Much less energy is really important. So I mentioned it takes about a volt to encode a bit in a computer, where the brain functions on millivolts, on difference of to 1,000 times smaller. So yeah, there are costs to everything, right? There's no free lunch, but nature seems to have gone for the solution that's redundant, lots of neurons, but robust to damage.
And how do you know that? So what are the sort of predictions which emerge from this sort of theory?
So whenever you have a redundant code, one of its property is that the neurons that are all encoding the same information should now be correlated, because they all encode the same thing. So they should share information. That's kind of what we mean by being correlated. And they have to be correlated in a very specific way. So it becomes a bit technical, but that's kind of where our work is focusing, is we have a very specific prediction about how the neurons should be correlated. And we figured out the mathematics behind that. And the bad surprise when we figured that out about 4 years ago is that our prediction had absolutely nothing to do with the data. So
so you mean that the data hadn't sort of matched the prediction, or?
Exactly. So we actually, people had measured already correlation before we did the mathematical work. And there's a long history behind that, but when the correlation was first measured, people declared, oh, this correlation implied that the brain has a redundant code. And about 10 years ago already, another group showed that, unfortunately, that's not the case. Given what we've measured in the brain, it doesn't look like a redundant code. So we came along and we said, yes, that's right. For a redundant code, we need another pattern of correlations. But the data was already published, and it didn't fit the data. So we had a real puzzle in front of us.
So how did you resolve it? So what's wrong? The data, your theory? How do you square the two?
So I'm a theoretician, so I love the data to be wrong. But the data is the data. So at the end of the day, what we came to realize is that the correlation that we see in real neurons are probably a mixture of different factors. Mm-hmm. Some of them that really constrain the code, makes it redundant, and others that are there because of the way the system is built. And we now have very good reason to believe that the measurements are completely dominated by the correlations that do not constrain the code, the ones that are irrelevant. So things like, you have different state of awareness, you go up and down, and we know today that this kind of fluctuation in the overall activity of the brain creates a particular pattern of correlations. Those turns out to have almost no impact on the code, but they create large correlation. Correlation are measured between 0 and 1, mm-hmm. and those are correlation coefficients, and their measurement shows about 0.2. The correlation that we care about, we could actually theoretically predict how big this should be. And they should be of about 0.01. So they are 10 times smaller than the kind of correlation that people measure. Mm-hmm. And correlations are notoriously difficult to measure too. In neuroscience, if we can get about 10 trials, 20 trials, it's a lot of trials. It's hard to get that from animals, and that's just not enough to know the value of the correlation coefficient. So we think that the correlation that we're looking for are currently buried under a lot of irrelevant fluctuation in the correlation coefficients.
So the correlation you're looking for is much, much tinier than these more irrelevant correlations, which just end up washing them out, as it were.
Exactly.
Obscuring them. So is there any way to peel them apart, to actually separate these two, to remove the sort of obscuring correlations, to actually get at the correlations you're interested in?
So we're not sure yet. We think it's going to be complicated, but there is hope, because sometimes we lose track of what's really we're interested in. Ultimately, what we're trying to do is to understand how neural activity controls behavior. Mm-hmm. That's really our final goal here. And the way we do this nowadays is we quantify how much information there is in your behavior. So how much information you capture in your behavior about the stimulus that is making you react, is making you behave. This we can quantify very precisely. And what we try to do is we try to see whether in the brain the information that we see in those different areas that are involved in behavior is indeed of the same order of magnitude, if not equal to what you see in the behavior of your animal or your subject. So we're trying to match information. So at the end of the day, what we want is not so much those correlations, which are kind of boring, counterintuitive, hard to measure. We want to know information. And so one way to do this is you measure the correlations, and from that you can compute the information. Another way is there are other techniques where you bypass the correlation measure altogether and you go straight for information. Now, the problem is that for doing that, we need to measure all the neurons at the same time while an animal is behaving.
I see. Yes.
So as you know, up until 10 years ago, that was a non-starter. All we could do, which was already remarkable, is record one neuron in one animal while the animal is performing a task. But we need thousands of neurons, and we know there are millions of neurons involved in any particular task. But what's fantastic in our field is there's been an explosion of technology, as you know, in the last 10 years, and we now are able to record at least from 100, possibly 1,000 neurons at the same time in a behaving animal. In fact, in some fishes, we can record all the neurons at once. So it's really amazing. So we can finally see the entire system at work live. And we have techniques now to try to figure out whether our tiny correlations are there or not by measuring information.
So can you, so again, this is very abstract work, but experimentally, what sort of techniques are you talking about? So again, for the audience who might be watching this sort of video, what sort of techniques would allow you to record from many neurons at once? So electrophysiology, for example, you're limited to recording from a few neurons. So what are the other sort of alternative techniques that you're looking at?
So yes, in the past, the way we used to record from neurons was by using basically electrical wires that we would stick into the brain, and we would push them until they would get very close to one neuron, and then we could actually hear the activity of that neurons and record its electrical activity. Nowadays, people have come up with absolutely stunning new techniques where instead they put dyes inside the cells, and they can have the dye expressed in all the cells at the same time. And those dyes are sensitive to an ion known as calcium. And whenever a cell is active, it's sending signals, calcium gets inside for a short time, for like 10 to 20 milliseconds. And the dyes that we express in those neurons, they can sense calcium. So they light up if they have calcium. So just by looking with light, we can then just look at how much light comes out, and we can figure out how much calcium gets into the cell, and therefore how active they are. So again, one has to keep in mind that we're talking about measuring things at the order of like a tenth of the width of a hair, and we're measuring now hundreds, if not thousands of them at the same time.
So, and finally, what sort of work are you planning in the future? What is over the horizon in the work that you use, the work you're doing?
So in the next few years, it's literally what I just described. That is, we finally have the technology, so we can finally now try to establish a precise mapping between the population activity of all the neurons together while the animal is performing a task. We can try to understand what it is in the activity of those neurons that leads to the behavior that we observe on the same trial. And that was, when I was a grad student, that was the dream, so that was already 20, 25 years ago. And I wasn't sure I would ever see a day where we would be able to do those experiments. But literally at this conference, I'm talking to colleagues here who have the technology, and that's the experiment we're gearing up to do. We're going to record all the neurons while the animal is performing the task, and we're going to use all the theoretical tools that we've developed while we were waiting for the technology to actually establish that mapping. So it's going to be really the first step toward really understanding this mapping between neural activity and or thoughts or behavior or desire or motivations.
Great. Well, I look forward to how things pan out. Thanks so much, Alex. That was great. Thank you.
Thank you very much.